Okay, so we are now live on this Friday afternoon here in continental Europe with another talk from the series from Open Neuroscience as part as the worldwide series. This is the open source track that is being organized by me and the other people organizing Open Neuroscience. And it's all about methods and tools around open source methods and tools in neurosciences. Today is my great pleasure to receive David and Matthew, who are the main developers of PiVR, which is a tool for closed loop behavior experiments with small animals. I'm not going to say much more about that because they're going to give the whole exclusive take on their project, but I would like to introduce them first. So starting with Matthew, he did his bachelor's in theoretical physics in the Free University of Brussels in Belgium. And in his PhD, before his PhD, he was a pre-doctoral fellow in the European Molecular Biology Laboratory at the European Bioinformatics Institute. He graduated in systems biology at the University of Cambridge, and his thesis focused on modeling the function of a gene regulatory network during Drosophila development. During his thesis, he became interested in the function of neural networks, and he joined the laboratory of Leslie Vosshall at the Rockefeller University, where he studies the mechanisms underlying the detection of olfactory signals in the Drosophila larvae. After his postdoc, he became a junior group leader at the EMBL CRG Systems Biology Unity of the Center for Genomic Regulation in Barcelona. And he worked as an independent investigator, delineating how orientation decisions emerge from neural computations carried out by the larval brain. Since the summer of 2016, he's an assistant professor at the University of California, Santa Barbara. David is a PhD student in his lab, and he's combining genetics, electrophysiology, and behavior analysis to investigate how olfactory sensory neurons encode odors in the fruit fly larva. David developed basically PiVR to parallelize his behavior experiments and to give summer school students an opportunity to get hands-on experience with a state-of-the-art behavior assay presenting virtual realities. So without further ado, I would like to then give the space to Matthew and David to talk to us about their system. Thank you both for agreeing to do this, and thank you for agreeing to do this even though you are super early in Santa Barbara right now. Thank you very much. The floor is yours.
Thank you, Andre. We're very excited to be able to present PiVR, this tool that was really transformative in the research of our lab. So I hope you can see the slides. Yes, everything good? All right. So PiVR was really a tool that allowed us to move forward with a project that almost looked like the dream 10 years ago. So it was a project that involved creating virtual olfactory realities for the model organism that we're studying, which is the Drosophila larva. And when we had that vision and we tried to make it possible, it essentially took us 5 years of research and development to get there, to put a tool in place that would achieve it. And that was done in collaboration with the Janelia Research Campus, so it's a huge project. And the cost of that tool, that first a closed loop tracker that was combining optogenetics and the analysis of behavior in real time, was $100,000. So there's a very expensive tool, and it became central in the research of our lab. So it's such an expensive tool created constraints because we only had one copy and we wanted to run many experiments with it. And there, David Tadres, who was starting his PhD in the lab, took on this challenge of trying to create a tool with the same basic functionality, but at a fraction of its original price. So making it possible to have many different copies of the tracker in the lab that was available to different lab members. But then on top of it, like we were able to bring this tool to summer schools and make it useful for educational purposes. So what we'll do in this presentation is to give you an overview of the genesis, the reason why we developed this closed loop tracking technology, how the PiVR tracker, so the closed loop tracker on a budget, operates, like why we think it's a really excellent tool, and also what's coming next. So what we would like it to be in the future, and where we think it may be a useful tool for you. So I'd like to start with the acknowledgement, because many different people contributed to the development of PiVR. So we had a lot of inspiration from the open source community. So the lab of Tom Baden with FlyPy, and Andre who's here. The otoscope from Giorgio Gilestro. And also I'd like to thank Vivek Jaraman, who helped me develop the first version of the tracker. Also, we had like a very strong incentive to make this tool affordable, to bring it to several summer schools. So one of them was the Drosophila Neurobiology course that's run at Cold Spring Harbor. And so various people contributed there, the course directors, you have their names on the slide, so we thank them a lot. And all our lab members who contributed in different ways to the development of PiVR. Also, they were beta users, but they went certainly beyond that in proposing many ways that led to improvement of the tool. I'm very proud that different various undergrads contributed to the use of PiVR, and they've been led by Nitesh Saxena, who's a postdoc in the lab. So all the undergrads have that star after their name, so there's quite a few of them. And finally, I'm very happy to be able to present PiVR with David, who was the person who had the vision behind making this tool affordable. At one point, it looked like a mission impossible. So when everything started in 6 years, and I'd like to just say that I think enthusiasm and creativity can really break nearly any technological barrier. So that's something that David did, and I'm very happy that we can share this presentation together. So let's start with the biological problem, because I think that explains what's behind the development of the technology. So we studied the Drosophila larva, and so that's this stage before the Drosophila becomes an adult fly that can walk and fly. So here the larva is crawling. It's about 3 to 4 millimeter long at the stage that we're studying. And during 6 days, a larva has to do something incredible, which is to increase its body mass by more than 100 times. So what it has to do is to feed constantly. And of course, feeding means that it has to find food. And to find food, it's using many different sensory cues, and among them, odors are critical. And the behavior that you'll observe here is driven by an airborne odor. So you have the odor source, which is that droplet that's yellow, that's hanging on the top of the ceiling, if you like, of a small arena. And the larva is starting at the opposite end of the odor gradient that will be created by that odor source. And here's the behavior of the larva. So the larva cannot see the gradient, obviously. It's just something that it can detect through its olfactory system. You have the head of the larva, that's that tiny black dot. And you see that that black dot is moving around and guiding the larva in the odor gradient. The remarkable thing is that the behavior is extremely well directed, and it is directed to a fairly complex process that involves searches, where the head is scanning the gradient, and of course, like movement of the animal into the gradient. And so fairly quickly, the larva reaches the peak of the odor gradient, where it's expecting to find food. Now, odors are difficult to work with, so it's a notorious problem. Their diffusion cannot be controlled so well. They're sticky, so they can contaminate the assay. And in spite of that, we were able to create good correlations between the sensory input and the behavioral output, which is a necessity if you want to understand the rules which are controlling behavior. But a few years ago, optogenetics just changed all those constraints because it allowed us to move away from odors and replace odors by light stimulation. So if you use a light-gated ion channel, like channel rhodopsin here, and you express this channel in an olfactory sensory neuron. So here we picked one out of the 21 olfactory sensory neurons in the peripheral olfactory system of the larva. So you express that channel, light-gated channel in that olfactory sensory neuron. When you shine light, you'll be able to evoke spikes in that neuron, which are triggered by light. And those spikes will be interpreted by the larva as if they were generated, produced by an odor. So what you can do here is to make the larva smell light in a sense. Okay? So the larva, of course, could respond to light, but we're testing larvae which are blind. So the larva is not responding to light per se. What it's responding to is the perception of odor that's induced by this optogenetic trick. Okay? So that's the basis of the technology that's behind the Closedloop tracker to create virtual olfactory reality. So if you now have a static light gradient that's shown on a substrate, so you have a light source and then some kind of gradient that's projected there. If you introduce a larva in that gradient, the larva will find its way to the peak of the gradient as if it were a real odor gradient, except that it's a virtual odor gradient because it's light-based. So this experiment was done in 2011 by Stortkohl and Fiala, for instance. I mean, we tried this as well, but we wanted to go beyond it. And why? Because with this kind of static gradient, even though the gradient is reproducible, its geometry is essentially fixed. So we wanted to have full control over the geometry, and we wanted to be able also to change the gradient over time, its properties. And so what we came up with in collaboration with Janelia Research Campus and Vivek Jaraman was this big, amazing tracker, which is essentially following the movement of the larva. So the larva is the tiny dot that you have here. It's moving on a stage that's 40 by 40 centimeters. And to keep a high resolution of the larva, so we maintain it in the middle of the field of view of the camera. And to do that, the camera has to follow the larva. So the camera is on a moving stage. And on that moving stage, you have LEDs, which are pointing, creating a spotlight that can excite the larva, so it can excite the neurons, in this case, the olfactory sensory neurons, which are expressing channel rhodopsin. So it is not that with this system, we're projecting a light gradient that has a certain geometry. What we do is that we just have the larva that's illuminated by a small spot of light, and then we change the light intensity of that spot of light. So I'll return to this. Now, this tracker that was developed in 2010, 2012, run with a loop cycle time that was fairly short, so 33 milliseconds. And that was very important to be able to update the light intensity as a function of the behavior of the larva. The resolution was very good because of the fact that the camera was following the larva. So you have approximately 100 pixels per millimeter. So a lot of information about the behavior of the larva can be captured. Then on top of it, we could do online behavioral classification. Okay, so that's the tool. And what you can do with this tool is create that light gradient with an exponential shape. And this light gradient is just based on the update of the light intensity as the larva is moving in that real physical space. So we have that predefined landscape, and based on where the larva is in the landscape, we just update the light intensity. And what you observe here is that the larva gets to the peak of that virtual odor gradient, will overshoot it because it's not finding any food source there, and then will return to it by realizing it's moving down the gradient. So you have that kind of dance around the peak of the gradient where the larva is overshooting it, returning to it. And that gives us a lot of data to try to understand what is directing elementary decisions like stopping and turning maneuvers. So this has been transformative in our research. And what it led to, for instance, is the ability to test models about the control of, for instance, here, lambda, which is the probability of stopping. So what we wanted to be able to do is to make predictions about when the larva is stopping to reorient. And the model that we're using is a probabilistic model, and the model is based on what is written here, the OSN, which is the firing rate of the olfactory sensory neuron. Now, the beauty with it is that the firing rate of the olfactory sensory neuron is controlled by light, by using our optogenetic trick. So we have full control over that, and we can then make predictions about which change in the activity of the olfactory sensory neuron will modulate the probability of stopping. And one basic model that we have here is, for instance, a model where we're testing like the modulation of the probability of stopping, where you have a maximum probability of stopping that's simply modulated by the activity of the olfactory sensory neuron. Yes, you have that proportionality term, which is gamma and like nothing more. And what we can do is create landscapes which have certain properties. So for instance, here it's a sort of volcano, and our predictions was that the larva would follow the rim of the volcano by zigzagging there. And you see that the color of the larva when it's becoming red is our prediction that it's going to stop and turn. And there's a relatively good correlation between our predictions and the behavior of the actual larva. And so this was done with a geometry that of an order gradient that you would not be able to create with a real physical gradient. Here you have an extreme version of it, which is a well, and whenever the larva is poking its head inside the well, there's no order anymore, so you have that abrupt change, and as you can see, the larva will never enter the well. So this gives you a sense of like how much optogenetics and closed loop tracking is allowing, like giving us in terms of making predictions about a model and predictions which are experimentally testable. So this tool, the closed loop tracker, was built in 2012. The price was about $100,000 with the research and development. And now the question was, can we make it cheaper? And one reason why the original tool was very expensive is that the camera had to move in such a way that we would keep enough resolution to capture the behavior of the larva. And also back then, simply purchasing LEDs, which were high intensity LEDs, was expensive. So one of the LEDs that you see here was costing, I think, $500 or more just there. So things changed in the meantime. And in particular, I think what changed was this barrier that became much lower in terms of creating amazing tools for not so much money by leveraging on technological development in the field, in particular making use of, for instance, Raspberry Pi microcomputer. And that's like where we had this very strong inspiration from the flyby, so the €100 lab tool. And so we started thinking like, could we actually make use of the same type of technology to create a closed loop tracker that would essentially do the same or nearly the same as our expensive tracker. Another source of inspiration came from the etoscope, and you can see here that if you have a behavioral assay that's based on Lego, so it's this combination of like having 3D printing and trying to simplify the design of a complex assay. And as I said, a big incentive for us became to bring our technology, the closed loop tracking, to summer schools. And like the ground where this took place was the Cold Spring Harbor Neurobiology of Drosophila course. And with various lab members and eventually David, who is here, we had iterations of the tracker. So you can see that like the first iteration, 2015, was essentially just a static camera. So of course that made everything less expensive. And then we had that static side LEDs. So it was capable of like producing a light gradient, have the larva respond to it, but it was not very flexible. And now here you have like the first version of the tracker that we had, which was 3D printed with that side LED red, looks a bit like the eye of the Terminator. The side illumination for the tracking was done, so you can see that ring that was suboptimal. Then we had to keep redesigning it. And 2 years later, David developed PiVR, and PiVR solved all those problems in amazing ways. So you have the Raspberry Pi technology for the tracking. David, using Python, created a fantastic GUI. The Illuminator is based on that platform. So the camera is static, and that was important to reduce the cost. And what you have now is a platform that's illuminated in the infrared so that you can do the tracking of the larva, and that platform is homogeneous. But what you have on top of the infrared for tracking is an LED, like a bunch of LEDs that can blue or red, that will create a homogeneous background that will stimulate the larva through the optogenetic gating of the activity of our olfactory sensory neuron. So what we have is that homogeneous background on which the larva is scrolling, and that background can have its intensity that's changing. So this is illustrated here. Now it's with an adult fly. The adult fly is moving in a checkerboard. So you see in the center, you have the predefined landscape, and the fly is moving, and on the left, you see that the light goes on and off depending on where the adult fly is. And so you have the change in light intensity that's shown on the right-hand side on that graph. So that's essentially the experiment as it's conducted. Of course, here it's like a change that's all or none, but you can create a greater change like for exponential virtual order gradient. Okay, so you have the freedom of creating any geometry that you may want. And I'll hand over to David here, who will tell you more about how to build PiVR, and more importantly, how PiVR operates.
Thank you, Matthieu.
Yes.
Can you give me, thank you.
Are.
Okay, so I'm gonna tell you in the next couple of slides how you can build one of these PiVR setups, and all the information you need is on this website, pivr.org. This is the landing page, and you will find the information of how to build a setup in great detail. You can, after you've built your setup, you can find a step-by-step experimental guide on typical pitfalls that you might run into if you just use it. And finally, there's also an in-depth software manual. You can even find the source code. All right, so as Matthieu said, the tool is quite affordable at $400 US, and it's quite straightforward to build. I usually start with the 3D printed part. So as you can see on the right, you will have to print a total 7 parts, and on the left you can see a time lapse of me printing exactly these pieces. So I usually start with this because this does take quite a bit of time, and we use a dual extrusion 3D printer because we like to have the support material, so that's the yellowish or whitish stuff here, to better print, for example, the casing, to have just better quality prints. So once this is done, to the next slide. So next we go to the soldering. So this might, if you've never soldered before, this might look a bit scary, but let me assure you, it's really not hard. I never had like formal training in soldering. So what you have to do is you go to the pivr.org website, you ask a company, you download this file, you ask a company to print it, and they'll send you exactly this for like a small price. So this is part of the $400 US price tag. And then what you have to do is to take your soldering iron and put all these different parts on the PCB. It's really quite straightforward. Even if you've never done it before, it's very hard to, well, we never had anyone break anything in the summer school we did, or yeah. So that's, I can only say it's really easy to do. Don't be scared of that. Just get a good soldering iron. And next, once you have the 3D printed part and the LED controller, the PCB that we built in the last slide, we can start putting everything together. And you can start with the monitor, which you can buy from one of the many suppliers. And as you can see, first you put in the casing on top of the monitor with a few screws. And then you can add the next step is to add the Raspberry Pi itself. And finally, we put in the LED controller that was soldered together in the previous slide. And there are two more wires essentially that need to be connected, and that's it. So now you have the most complex part of the PiVR setup is built. So all that's left is now the tower that contains the camera. And as you can see, I'm wearing gloves here, and that is because there's an infrared filter on the camera itself. So I'm just wearing gloves not to have my fingerprints all over the optical components of the setup. And I hope you can appreciate that it's really all, it's a bit like Lego, where we try to have as few steps of putting the whole setup together as possible. There are a few screws that need to go in, but that's it. So you see now the tower is connected to the casing itself, and we are now completely done with that part of the setup. So all that's left is to plug it in, and if everything's put together properly, your PiVR setup will actually start up. So there's one more piece, the illuminator that Matthieu was referring to before. Yes?
Sorry, let me just, sorry to interrupt you, but I had a question because you mentioned that you're using a dual extrusion printer. Because of the better quality. Does it also work if you don't have one of those? Or
we haven't tried.
You recommend it? Okay.
I would recommend to use the, okay. I think it's gonna not, it might work if you're lucky. I don't know if it's gonna work. Let me know if it works. Let me know. You can shoot me an email.
Okay. So the reason why I'm asking is also because we recently got one of these Creality printers, which are quite inexpensive, and we've been like putting everything on it to see if we can print as good, and the prints are quite good actually, which was quite surprising in terms of the time that it took it to tune it in and to get good prints. And so, because we know that a lot of our colleagues that are connected via the Trend in Africa network have one of those. And so I've been testing like what works and what doesn't, you know? And so, yeah, that's why I'm asking. Sorry, I don't want to take more time, but then like now on my to-do list is to print one of those and see how well they come out and let you know.
Yeah, yes, please do. And I think there's probably this, I think this 3D print file probably can be simplified. So also if you have any suggestion or anyone in the audience has any suggestion of how to improve it so that the file, so that it can be printed without the dual extrusion system, I think that would increase the reach of PiVR.
Yeah, I don't think there's any part that requires precision printing. What's possible is that the current design may not work perfectly if you don't have enough precision, but as David said, the simplification would be probably relatively straightforward. So do get in touch with us if it's working for you, and if it's not, I think we can help you make it work, and we would definitely want to make sure that the PiVR design is compatible with different types of printers. So thanks, Andre, for this question. I think it's certainly an important component of the tool building.
It just go back the slide. Okay, so here we are. Okay, so here we have the last part that needs to be built is the illuminator. So that's literally just LEDs on top of plastic with a diffuser on top. So as you can see here, we have two different colored LED stripes. So one of them is going to be the infrared that's used to detect the animal with the camera. And the other one is the, in our case here, red light to stimulate crimson, the optogenetic tool. And finally, there's a cable that you can make yourself to connect the illuminator to the PiVR setup from the last slide. All right, so now the installation is really, really straightforward. You go to the website, you download a script, and you enter one line in the terminal, and you hit enter, and it does everything itself. It even restarts, and you can then use your PiVR setup for the first time and run some tests as described on the website. So so I've just shown you that it's straightforward to build this setup. or as straightforward as it can be. So one very important point I want to highlight today, and maybe the take home, like one of the main take home messages for people who think about maybe using PiVR. PiVR has a high frame rate and a low latency. So I'm going to go into this in a second, but you might first let me explain why this is important. So you might think, well, you guys are studying larvae. I mean, larvae are not known to like be super fast. So why do you need high frame rates and low latencies, right? So let me show you a video on the next slide. So what you can see here on the top is the actual larva, and this is the binary image. At the bottom, you see the time, how fast everything moves. And let me play this again, but probably it's hard to see anything, right? So let's slow this down. This was real speed. Let's slow this down 10%. And you can see here, this is the head or nose of the animal, and it's moving to the right and it's moving to the left. And all this happened, as you can see here, in less than 1 second. So if you want to capture this head cast, that's what we call these lateral head movements. If you want to capture these head cast dynamics, you need to know how fast are they moving. They're moving surprisingly fast. They move at more than 3 millimeters per second. And a head cast can be over in 1 to 200 milliseconds. So if you want to study these kinds of dynamics in the larva, you need to have at least 10 frames per second. Probably it's better if you have 20 frames per second. This is also where, as Matthieu described before, the 30 frames per second in the original tracker, that's why these were necessary. So this is why we need to have a high frame rate, and depending on the model organism, or depending on the organism you're using, you probably know better how high the frame rate needs to be to study the behavior you're interested in. So second is the latency, and I think it's not always completely clear what the difference between frame rate and latency is. Yes, go ahead.
Sorry, before you start the latency part, there is a question from the audience. I'm not sure I'm going to pronounce her name correctly. Hagenhild? Irene, maybe. I don't know what she likes to be, what she goes by, but she's asking, what is the resolution of optical stimulation through the diffuser? Is it possible to move the stimulation LEDs closer to the top diffuser plate to increase the spatial resolution?
So you don't have, and like so you need to think back at the video that Matthieu showed, where it's an all or none, right? The spatial resolution comes by the time resolution. So you, depending on where you are, or maybe another way to think, okay, I see, maybe I misunderstood the question. So the resolution that you have is how many pixels you have in your camera, essentially. So if you have 640 times 480 image, this is your resolution of how precise the light can be stimulated. So I hope this. So if you increase the camera resolution, then you will increase the resolution of the stimulus. Does that make sense? So otherwise, we can show the video again that Matthieu showed on his last slide.
Yeah, maybe this question you can specify it. I think if it's just like the resolution of the tracking itself, of course you can increase it by bringing the camera closer to the animal, but I'm not entirely sure what you mean by the resolution at the level of the optogenetic stimulation.
Irene, do you want to unmute yourself and tell us if this solves your question? Sorry.
Or maybe we can revisit this at the end of the talk.
Yeah, because I think she unmuted herself, but I could not hear her.
All right, so perhaps let's revisit this question later.
Okay, and
yeah,
if she wants to write it down here on the chat, then we can also have more details for the end of the talk.
Mm-hmm. And David has more data about like what's happening when you use cameras with higher resolution. So you'll see that at the end of the talk.
Yeah, yeah. So if we don't answer a question during the talk, please come back to us. All right, so as I described in the previous slide, we actually do need a high frame rate. So what's the difference between frame rate and latency? So latency in our case, if you remember, the animal moved its head to the right and then to the left in around 100 milliseconds. Latency is the time between the movement of the animal and the actuation of the LED. So for example, if we want to turn on the LED every time the animal moves the head to the right, but if the latency was 1 second, the animal would already have moved on and the light would only be on much too late. another way to think about this, if you have these modern virtual reality headsets for humans, imagine if whenever you move your head to the left and then to the right, the image updates always 1 second late. I haven't tried this. I assume you will get very bad motion sickness. So this is what latency is. And so we need both high frame rate and low latency. So how does PiVR perform? Obviously, it depends on the resolution that we're using. So here I just jotted down 3 resolutions, low, medium, and high. And at low resolutions, you can achieve more than 60 frames per second. And at high resolution, what we call high resolution, at 1296 times 976 pixels, you get still more than 30 frames per second. So really the loop time is in the order of tens of milliseconds. So for us, this is really all we need. For larva, actually 30 frames are probably enough for most of the experiments. And so at the bottom, you can see the comparison of like how does the image look of these different resolutions. So on the left, you have the low resolution, on the right, you have the higher resolution. And I think you can appreciate that the higher the resolution, the less pixelated your image is going to look. But I want to highlight that for the paper, everything was done with the 640 times 480. So if you just need to find your centroid, this is completely fine. If you want to have, if you need better information about which part of the animal is where in the image, then you probably want to increase your resolution. And regarding the latency, so again, this is the time between the image being taken until in our case, the LED is being turned on. We measured this and it's less than 30 milliseconds, which is pretty fast. So yeah, so just these are the numbers really. So we have 60 Hertz and less than 30 millisecond latency. So now you might be wondering, well, how is this possible? These guys are only using this tiny computer. So how is it possible that you have this high frame rate and this low latency? And here, I think I really have, and I really want to, and I'm happy to do so, is to give a big shout out to the open source community. Without them, like PiVR definitely would not have been possible. So it starts, of course, with the operating system, Linux, the most famous open source project, I think. Then you have the programming language, Python, which is, again, it's completely open source. Python has these packages, which are, again, provided by people. Usually they're provided open source, and everything you can use here is free. So for example, a very important package, which you probably already used if you ever use Python, is numerical Python, and PiVR uses it for image processing as it allows for really fast matrix operations. And then 2 more packages, but PiVR actually has many more, but I can't talk about all of them. It's the PiCamera package, which allows me to very easily get access to the camera in Python. And finally, the hardware controlled is PiGPIO library, which allows me to directly control these general purpose input and output pins with a microsecond precision. So how does the tracking algorithm work? So I think one important aspect of the tracking software to understand is that it never really works. That's really the trick why it's fast. It never really works on the whole image. So if you have an image coming in from the camera, it's immediately, before anything is done really, it's immediately cropped to a region of interest. So only a very small area. And you then subtract this small region of interest from the actual background image and you get a high contrast image. And then it's all pretty straightforward after that. So now you just, if you look at the histogram of the pixel intensity of this small image, of this subtracted image, you will see that you have a lot of zeros, which are the background here, and you will have a few pixels which are different from 0, and that's your animal you're interested in. So all you need to do is threshold, then you can binarize your image, and all the further image processing is done on this binarized image, and there are a lot of good tools to do that. So this is pretty well established. Once you have this image, you can easily find the centroid, skeletonize the whole image, etc. And then just to close the loop, so what PiVR then does, it saves only this tiny image. So not even the whole image I'm showing here, just the tiny image of the animal itself. It defines where the animal can be in the next frame. Because we have this really high frame rate, we can predict where the animal will be in the next frame because we know it cannot suddenly jump around. to the other side of the dish. So you know that in the next frame, you only have to look at this particular region of interest. And this whole loop, then you grab the next frame and the whole cycle starts again. And this whole loop at 60 frames obviously takes less than 16.6 milliseconds. And if you have higher resolution, of course, it will take a bit longer because you will have more pixels to work on. And that's the tracking algorithm. So now, one more technical aspect I want to go in before we go back to the biology is, I've told you what happens. So you, from the camera, the image is fed into the Raspberry Pi, the animal is tracked, as I've shown in the last couple of slides. But now the question is, well, how do you control the LEDs? And here, I want to quickly give a shout out to LEDs. LEDs are really awesome. You can turn them on and off in like nanoseconds. Like it's really crazy. And what we can use to control the intensity of the LED is pulse width modulation. So what PiVR does is, I hope you can see in blue these lines. Every 25 microseconds, you have one pulse. So like it's 40,000 hertz, 40,000 times per second. So at the bottom here on the x-axis, you can see that we have 100 microseconds. So not milliseconds, microseconds. And every 25 microseconds, we have one pulse. And all we do is we change the time at which the LED is on and off. And so for example, at 50%, if we want 50% light intensity, what PiVR will do, it will turn the LED on 12.5 microseconds long, then it will turn it off for 12.5 microseconds, then it will turn it back on. And let me just like be very clear here. So this is, the LED truly is on here and it truly is off here. But I mean, our eyes definitely can't see at 40,000 hertz. Like we just integrate over time. The larva definitely can't see at 40,000 hertz. The camera cannot detect the single on and off events. And for everything we know, like the channel rhodopsins, the optogenetic tools we're using integrate over longer time scales than 25 microseconds. So for all intents and purposes, the 50% light intensity equals 50% of light on and off. And of course, you can do everything in between. I only plotted 25 and 75% light intensity here, but you could have 13.2%, you can have 89.2, whatever you want. Anything in between works. So you have this complete digital control of the LEDs. So here I took a video where I'm just dragging the slider in the PiVR setup first so that the LED goes on and off, and it's designed to be like in this staircase pattern. You can see like first it goes up and then it goes down again in this duty cycle percentage. So essentially goes 5, 10, 20, etc. %. So now back to the biology. As Matthieu said, we express the optogenetic tool, in this case crimson, in the olfactory sensory system of the larva. And now this is data from PiVR. And what you can see is that we are presenting this Gaussian gradient that looks like, essentially mimics a real odor source. And the animal starts at the foot of this hill, of this Gaussian hill, and it quickly turns around and then it spends the rest of the time looking for the peak. So it looks very similar to what Matthieu showed before. And then here another example on the left, you have the donut-shaped or volcano-shaped virtual reality that Matthieu already showed before from the original tracker from 2013. And on the right, we did the same experiment, but with PiVR. And as you can see in both cases, the animal behaves the same. So I think PiVR really does perform on par, at least in these experiments that we tested as this original tool.
So hopefully we've convinced you that PiVR has really good performances to create virtual realities. And what I'd like to do now is to show you some of the applications that we've made with this tool that went beyond simply studying chemotaxis in the larva. The first application will actually illustrate that the tool is accessible to, I think, non-specialists. So that was like really one important element in its design. And so here we have a group of 3 undergrads worked in the lab to test one hypothesis, which was that the latest, most popular tool for optogenetics called Crimson. And so that light-gated ion channel is activated in the red range, and it comes as 2 versions. So you have the white type version that's called CS Crimson, and then you have another version that has been point mutated, and that produces an off kinetics which is faster, and so that's Crimson R. So you can actually see that, like, after having generated an action potential, you have the recovery that's faster for Crimson R. And as a result, when you try to create action potentials with pulses of light, and so that's what is shown in the top panel, the performance of Crimson R are more reliable than CS Crimson. So you see that, like, for pulses which are really fast, if you try to produce a relatively strong firing rate, you will manage to sustain it with Crimson R much more than with Crimson. So that was characterized in the original paper where Crimson was introduced in 2014 by Klaputke and Boyden. So what we decided to do was to express Crimson R and CS Crimson in that single olfactory sensory neuron to produce chemotaxis and ask whether each version was leading to the same level of chemotaxis in the same virtual order gradient. So we fixed the geometry of the order gradient as discussed before, and then we tested those different larvae. So the first one is the CS Crimson that has been used until now in the presentation. And you observe that you have accumulation near the peak of the virtual order gradient as expected. This is the control where you have Crimson, so the 2 versions of Crimson, but not driven in the, its expression not driven in the olfactory sensory neuron. What you observe is that even though they're starting close to the center, it just diffuse away from there. So you don't have chemotaxis, which is elicited. And now we have the performance of animals which are expressing Crimson R in their olfactory sensory neuron. And by comparing the graph that you have at the top for CS Crimson and the one at the bottom for Crimson R, I hope that you can appreciate that clearly, like, the Crimson R larvae are more accurate in the way they chemotax. So they appear to be able to detect that they moved on the gradient quicker, which allows them to turn around and return to the peak with a shorter latency. And that leads to this accumulation near the peak that's more precise. This has been quantified by the 3 undergrads by measuring the distance to the source for all the animals that were tested, 28 larvae, and then taking the average. And what you observe is that the Crimson R larvae are able to stay closer to the peak of the virtual order gradient. So being closer means that you are closer to 0. Then the CS Crimson is not as accurate, certainly not at the beginning. So they tend to leave the peak of the gradient and then they return to it. And then you have the control, which gives you that sort of like distance that larvae will tend to adopt when they cannot detect the virtual order gradient. So this is very interesting because the project here is linking the response properties of the light-gated ion channel, like different variants, to a macroscopic manifestation of changes in the firing rate of the olfactory sensory neuron, because chemotaxis, like what we're quantifying here, distance to source, is very much to do with the behavior itself. And so hopefully that gives you a sense of like how far you can go with like the use of PiVR to detect subtle differences in behavior. Here's an application with adult flies. So adult flies will walk faster than larvae crawl. So we were curious to see if we would be able to track them and then create virtual realities. And what we did is that we created a virtual reality where the fly would experience bitter taste when illuminated. So you have crimson, which is expressed in the bitter taste neurons. And so now the fly is standing in a quadrant that's not lit. So it's a dark quadrant. It's not detecting bitter taste. And now it just entered the lit quadrant. So it did that a couple of times. And it was strongly repelled because it's sensing bitter taste and it's trying to avoid it. So that was it was nice to reproduce these results, which were known in the field, but what PiVR allowed us to do was to explore like what leads to this aversion. So the first thing is here the quantification that shows that the adult fly will tend to spend less time on the lit quadrant when it can detect that virtual bitter taste. So the negative preference index that you have here is essentially showing that the adult fly will prefer the dark quadrants. And that's, I think, where PiVR was like very convenient in all the behavioral quantification. We are able to show that the flies, which were expressing crimson in the bitter taste neurons, when they were exposed to light, so when they were sensing bitter taste, they were accelerating. So essentially, this behavior is modulated by the fact that the locomotion speed tends to be low. The fly will even stop when it's not perceiving bitter taste. And when it's perceiving bitter taste, it will accelerate to try to find a region of space where it's not stimulated by the light anymore. It's not perceiving bitter taste. So that's like one sensorimotor rule, it's just like based on change in speed to produce an aversive response. Okay? So that's what we learned here with adult flies. And here's another even more challenging application that we had for PiVR. And so that led David to go back to zebrafish, which he worked with during his master thesis in Zurich. And the fish larvae are innately attracted by light. And so we created a light gradient. Now, what you have to imagine is that this light gradient is not associated with any form of stereo vision or depth perception. It's just light that's being shown on both eyes. So the only thing that the fish detects is a change in light intensity over time. And what we asked was whether this was enough for the fish to identify the peak of the light gradient and stay close to it. So you have the change in light intensity that's at the bottom and the behavior of the fish. Here you have the quantification of the behavior. So when you look at the speed of the fish, you have that periodic behavior, which is associated with bouts of motion. That's because the fish moves through the powerful movement of the tail, which will create like a swim bout. And then it will slow down, and at that point we'll be able to reorient. And you see that the light intensity is changing almost discretely whenever you have a swim bout, right? You have an increase or decrease in light intensity because the fish is moving relatively fast. And PiVR was capable of like tracking the fish and created a change in light intensity. And as you can see, when you have a light gradient, the fish will tend to stay at the center of the gradient. And of course, when there's no light gradient, they'll diffuse away from that region of space. And again, what we were able to ask, and that was a challenging part and also driven by curiosity, is like, how is the fish capable of doing this? Like, what's the rule, the sensorimotor rule, this conversion of sensation into action that would lead to positive phototaxis? And what we quantified here was the change in orientation, so the turn angle between swimming bouts. And we analyzed it, we correlated with the stimulus intensity. And the first thing that we found was that fishes which were experiencing that light gradient, virtual light gradient, because it's, there's no depth perception with it, would tend to turn more when the stimulus intensity was low. So it was like kind of, something similar to the adult fly. So the fish will turn more at low light intensity. But on top of it, and that was the really interesting element, is that you also have a modulation that's dependent on the change in light intensity that the fish is experiencing. So even at low light intensity, if the fish is moving up the gradient, so if it's experiencing an increase in light intensity, it is not going to turn much. So that's what you have for the green case, for the purple case, like when it's moving up the gradient, the turn angle is not that large. But if the fish is moving down the gradient and the light intensity is low, then it will turn more. So you have this sort of like end rule that's controlling part of the phototactic response of the fish. And there's a much more in-depth analysis of the sensorimotor control of phototaxis in zebrafish that was published in 2020 by Georges Dobreja. And we were actually quite excited to find that in his manuscript, some of the elements that we had observed with PiVR were confirmed and then like further expanded. So we really think that PiVR is a good tool for you to ask cutting edge questions with various small model organisms. And it's like not just zebrafish or Drosophila, like David tested the PiVR tracking on different insects. So you have here spiders and all sorts of things, and it works well. So if you wanted to study, for instance, phototaxis with these animals, you could. If you can re-engineer them with now CRISPR and have optogenetics expressing them, like you would be able to create also virtual sensory realities through optogenetics. Also, we tested leech, and this is like analyzing exploratory behavior in leech. So I think it's a very versatile tool. David will tell you a bit more now about like where the tool is going next, like how we can piggyback on the development of the Raspberry Pi microcomputer to do ever like better tracking and create better virtual realities.
Thank you. So everything or most of what we've shown you so far was done with this illuminator that I've already shown in the video before, that allows you to both illuminate the animal for the detection algorithm and stimulate it with red, blue, green, or whatever LED you want to use light for your optogenetic or visual stimulation. You don't get a lot of light power out of this normal, or depends on what you need. You get around 2 microwatts per square millimeter out of an illuminator like this. If this is not enough, PiVR allows you to use a high-powered LED, of course, so you can just use much higher powered LEDs and you will essentially jump the light intensity by an order of magnitude here. So you need a different LED controller. This is all described in the website. And finally, one, I think, very exciting aspect of preparing your own experimental devices is when 3D printing everything is that, I mean, you're completely free. In one case, we wanted to have side illumination. So we just printed this round case and we have the LEDs you can see here on the inside and you can see the petri dish at the side. So in this case, instead of the illumination coming from below, you have illumination from the side. And this helped us to get more details of the animals on top into the camera. And one really exciting aspect, I think, for many people could be that since PiVR is high frame rate, low latency, producing pulse width modulation, you can create virtual realities with things different than light. So for example, in preparation for this talk, on Monday, I spent 30 minutes, I spent 30 minutes and we were able to use this piezo to create a virtual sound reality. So you can see we have this fake animal here, and you can see here where it is relative to the virtual reality. So the more red, the more high intensity you want. And if it's not red, you don't want to have any virtual reality. The more this piezo should be making its noise. As you can hear, very unpleasant noise, but you can very easily, you can see now it's gonna turn off or turn less on. And so what we want to show here I'm sure this can be optimized and made much better, but I think what we want to show here and demonstrate here, that it's really easy to use other output devices than LEDs for your closed loop assay. And, Finally, as Matthieu said at the beginning, there were two goals for the development of PiVR. One was, yes?
But there is a question from Panos. What are some types of non-Gaussian gradients particularly challenging to generate, but possibly common in nature, or otherwise valuable in the context of some neuroscience experiment?
So I think, so for example, what we've shown before was the donuts. I mean, obviously this will not happen in nature, so the animal does not need to be especially good at detecting it, right? But they are very good at staying on the rim. And what this tells us, so if we have an hypothesis of how you have the sensory motor transformation happening from the neuron to the behavior, if we have a model, we can test it. And that's, I think, where we are using virtual reality a lot, is in testing unrealistic models. So it's really going into the opposite of the question, probably. If the question is, well, why would you need to create virtual realities which are unrealistic? We're coming from it really from the other side. It's like, well, we see the behavior. We have a model of how the animal should have integrated its environmental information into behavior. Therefore, if we manipulate the environment in such a way, we predict that we get this particular behavior. So this is what we are mainly using virtual reality for. Do you want to add something, Matthieu?
No, I think it's the animal doesn't really know like how that gradient looks like. It's responding to it. We can make predictions about how we expect it to respond. If we were to find a condition that the animal cannot cope with, like a sort of geometry, or even like if you were to introduce perturbations that it would make the animal unable to chemotax, I would say this would be like a very, very interesting condition, because it would show that we're pushing the system to a limit where there's a breaking point, and that would certainly tell us about the design and the purpose of that system. So perhaps like if you have certain conditions which are unrealistic and the animal cannot cope with them, it would tell you something about the ecology of that animal. So I think it's very much complementary. But what is true is that those virtual realities, you're trying to capture what we think is the essence of what an animal is detecting, and you play it to this animal and you see whether your model is appropriate or not. So I think that's just no, you can also create just static order gradient and track your animal and test its behavior there if you want to have something that's much more naturalistic.
Thank you. Excuse me. So as Matthieu said before, there were really two goals when we developed PiVR. One was that we can bring state-of-the-art, relatively affordable and easy to build setups to these summer schools so that the students have access to a state-of-the-art virtual reality system. The second one was that I, as a PhD student, I want to run more experiments. And I think what you can appreciate here is that what I'm using them all the time now for all my tracking needs, for all my behavior observation needs. So I'm using this little farm here that you can see here. This is one of the farms we have of four PiVR setups. So I, as a PhD student, can run 4 times as many experiments with this setup as I could with the one tracker. So that's really helpful. So you can run screens much faster, for example, if you have more tools, obviously. Now I'm gonna next a couple of slides talk a little bit about the environment where PiVR is and why we think it's a very safe environment to be in. So, so in 2015, the Raspberry Pi 2 was released, and we started shortly thereafter. We started developing PiVR, and in 2018, the Raspberry Pi 3B Plus came out, much more powerful computer than the Raspberry Pi 2, and this really allowed us to. The Raspberry Pi 3 really allowed us to do everything closed loop for you because it was just fast enough. And then the most recent iteration came out in 2019, it's the Raspberry Pi 4. Why am I showing you this? Well, it has the CPU power, has direct implications on how many frames can process per second. So you can see here in dark green, especially at the high resolution, you get a doubling of the process frames from the Raspberry Pi 4 relative to the Raspberry Pi 3B Plus. And if you check the numbers up here, it's very likely that we'll get a Raspberry Pi 5 and then like next year, maybe the year after. And looking back, it's reasonable to project that we'll again get a proper bump in the frame rates that can be processed by this new, more powerful Raspberry Pi. And finally, a very interesting, another interesting aspect of the Raspberry Pi environment is that so far we've done everything with this really tiny camera. The Raspberry Pi Foundation released a high quality camera, and I think there's a big brother of this little one here. And it's not only much bigger, what's very important is the sensor, this silicon in the middle here is much bigger. So here, I think you have like a 10% bigger pixel size in the high quality camera compared to the original one. Of course, the optics is also much, much better. As you can see here, the objective is orders of magnitude bigger than the small one. And just to give you an idea, so this is an image taken at 30 frames per second with PiVR. And I think you can appreciate, you do see more details. So I don't know if you've noticed at the beginning, I'm going to play it again. The animal don't only see that it's doing the lateral head cast, you can actually see how it's lifting the head. So the animal is not only like moving in 2D, like everything we've shown you so far. With this camera, in principle, it's possible to, it's possible to study the behavior in 3D. This again. So you can see how now it's moving the head, and now it's moving to the right. And this is slowed down to 25% of the original speed. Otherwise, again, it would be very hard to see anything. The other advantage you could use, the other advantage you have by using a high quality camera is to still be like, use very low resolution to go very fast. You still get a better image, of course, because you just get more information in the camera. And finally, I want to close with this slide. We've been using PiVR for several years now. Especially in the last year, we have quite a few people who started using PiVR in our lab, new postdocs, new graduate students, a lot of undergrads are using it. We know that other labs are using PiVR. So we're at this point pretty confident that the hardware works and the tracking algorithm works as it's expected to. So we think the next step for us is to help the community with the data quantification that's coming out of the PiVR setup. And I think this is now an invitation. If you think you're interested in using such a data quantification package, please shoot Matthieu an email, because we would like to better understand what your needs are to help you analyze your PiVR data. Thank you.
Thank you both for this amazing talk. I would like to ask people here in the Zoom room or on YouTube if you have any further questions. I think now it's a good time to make them. If you guys don't mind, I actually would like to kick off with one of mine. So you were saying in the beginning how the animals go to the center of the gradient and then they don't actually find food and then they overshoot and go back again. Were there any experiments where you actually try to have there, where the center of the gradient is like a little food sample or whatever it is that would be like a positive feedback loop? Because in principle, you could add like a 2-axis motor and just have, you know, like this food sample moving around together with your gradient, no? And so the animal would have like this, okay, I follow the camels, I followed this with the sense of smell and I actually found what I was looking for. Would this bring any difference, you think, to your experimental results?
Yeah, so you could probably try to do that. I'm not entirely sure that you would be able to move food that would provide reward and without leaving a trace. And if you really wanted to do that, you would probably go for optogenetics in maybe like trying to combine blue light activating reward neurons with red light activating the olfactory system or something like that, such a way that you could make it conditional that when the larva is reaching the peak of the gradient, suddenly it would be rewarded for that. So you could imagine an experiment like that. When we conducted these experiments with the larva getting to the peak of the gradient, not finding food, we were actually interested in having them search for much longer and like return to the peak and then drift away. And And the reason for that is that we were focused on understanding what is making them stop and turn and how they're capable of turning. So in fact, having many of these instances and not having the larva stop at the center and be happy and content at the center was like what we were trying to do. But for sure, there's a big interest in the field in understanding how the internal state is affecting it's called sensorimotor integration, sensorimotor transformation. So there, for sure, like having some, say, like sugar or like some kind of yeast or detected at the center would be something interesting. In fact, I mean, some of these experiments are being done. I mean, Jimena Berni had recently a very nice bioarchive preprint where they sort of try to address this question.
Great, thanks. There are no questions from the audience, but maybe it would be a good time to go back to Irene's question. I don't know if this was solved, Irene. Do you want to maybe let us know if during the talk, the question that you had got solved, or if what by she wrote on the chat here on the Zoom room, if you guys get a better idea of what she was asking? So basically, okay, so she says it's fine. I think she misunderstood the setup at first. Okay,
okay.
So thank you for that. I'm glad to
yeah,
hear
there's
that
maybe one more
clear.
point that
Yeah.
I'd like to make here in closing is that I think the approach we took with PiVR and like all the closed loop tracking technology was, I think, inspiring, like in many ways for our lab, which was going from a complex, sophisticated research tool, so that expensive tracker, and trying to simplify it because we wanted to have more copies in the lab, but also with the challenge of bringing it to summer school, so have an educational purpose in it. And one may have seen this, at least like the educational component, the science outreach, as perhaps it's like not completely artificial, but like, can it really help you in your research? And the fact that we wanted to make it open source and document it very well, and like, is this not like just some kind of time that's taken and wasted from your research? I think the answer is absolutely not, because all the challenges that we had to solve technically, and that was really what David did over and over, they helped us create a tool that ended up being much better for our own use. So it simplified what could be simplified. It made it more modular. It made it more flexible and versatile. And I think that's how it created a tool that we really hope that the community can also benefit from. So time will tell whether that's the case. At least, like, I think for our own lab, it has been very successful. And we hope that we'll see more of this, because it's probably the best way also for us to be able to compare data for experiments which are relatively similar. I think if we use tools which are, more or less the same, probably being able to interpret results like generated by other labs will be easier in the future.
Okay, thanks. That's a very good point as well, that it's not a waste of time, and then you get all these benefits from, like I'm just trying to follow up on what you're saying, that what initially people might think it's a waste of time, it was really useful for your lab and hopefully for others, right? Are there any other questions from the audience? I think we are reaching the point where it's speak now or forever. Hold your peace, or actually contact the author like David and Matthew later on Twitter or via email, or from whatever communication method you can find. If that is not the case, I would like to say thank you again. Irene is also thanking you for your presentation.
Thank you, Andre.
Yeah, you're welcome. Umesh is also saying thank you. I mean, I guess you can see on the Zoom chat, but I'm just saying because the people on YouTube are not seeing the chat, so I would just like to give them the feedback as well, the live feedback for everyone. And with that, I think we can close the YouTube transmission in a couple of seconds. And if anybody wants to hang out a little bit longer and ask people and chat and ask people things, they can also do that. That's absolutely fine. And with that, I will end the live transmission on YouTube. which is the